Hello, my name is Gabrielle Heltzel. I'll be covering an interesting topic on the current evolution of how our bodies handle alcohol and how evolution may make it harder for humans to hold their liquor. So, how long have humans been consuming alcohol? The history of alcohol and humans is at least dated back 30,000 years and arguably even longer than. It was made and consumed by the prehistoric societies in 6 of the 7 continents in varieties of forms based on a variety of natural sugars found in grains and fruits. Why did I choose this topic? I chose this topic because it was a very interesting title. I had seen the studies and conducted. Personally, I can relate because I have numerous friends that have the flushing and they metabolize alcohol differently. A lot of aspects about the modern human condition goes back to our evolutionary history, and the ability to break down alcohol likely helped human ancestors make the most out of rotting fermented fruit that fell onto the forest floor. I think it's a very interesting topic because it is dated so long within our own history of how we've consumed alcohol. So you can see on the picture to the left, it kind of just shows all the different particular forms of alcohol that we do consume nowadays and how far it was dated by the first production of this particular form of alcohol. The study that we will be reviewing today was originally conducted to determine alcohol dependency. The results have even more interesting findings about the geographic and evolutionary systems of how we metabolize alcohol, but we will get into the results later. Recent positive selection has been a driving force in human evolution, and the studies of loci targeted by positive selection have uncovered potential additive phenotypes in recent human evolutionary history. Vaught, who's the scientist who had conducted this research, said that they did the study so they could learn which regions of human genome have adapted and evolved over the past tens of thousands of years. To investigate this, they looked at publicly available data from the Thousand Genomes Project, which is a large sequencing venture that collected genomes of more than 2,500 individuals of diverse ancestries, representing 26 different populations from 4 continents. Signatures of recent positive selection often overlap across human populations, but the question of how often these overlaps represent a single ancestry event remains unresolved. If a single selection event spread across many populations, the same sweeping haplotype should appear in each population, and the selected pressure could be a common across populations and environments. Identifying such shared selective events could identify genome loci and the human traits important in recent history across the globe. In addition, genome annotations that recently became available could help attach these signatures to a potential gene and molecular phenotype selected across populations. In this study, they have presented a catalog of selective sweeps in humans and identified those that overlap and shared a sweeping haplotype. They then connected these sweep overlaps with potential biological mechanisms at several loci. Current known adaptations of alcohol. When people drink alcohol, their bodies break it down into a toxic intermediate known as acetaldehyde. When acetaldehyde accumulates in the body, it can lead to adverse reactions, include facial flushing, nausea, and rapid heartbeat. This is according to a 2007 report in the Journal of Alcohol Research Current Reviews. Also, some people with the East Asian ancestry have a genetic variation that makes it uncomfortable to drink too much alcohol. This variation reduces the function of the gene that converts acetaldehyde into the less toxic acetate. So moving forward to show the studies conducted in this research study, I will be presenting the catalog of the selective sweeps. In figure 1 on the left-hand side, this is the normalizing of IHS by local recombination rate. The mean is the solid line and the variance is the dashed line of the IHS as a function of local recombination rate. IHS values normalized by derived allele frequency are shown in green. Normalization by both derived allele frequencies and local recombination rate is shown in purple. The gray lines represent the mean variance for comparison. And then figure 2 on the right-hand side is shown the closely relative populations with sweep overlaps more frequently. For each population pair, the fraction of sweep intervals that overlap is plotted against pairwise estimate FST. Each population pair, or the dots, is colored by its continent groupings. For example, EUR versus SAS is one European population versus one South Asian population. AFR is African, EAS is South Asian, EUR is European, and SAS is Asian. In this graph, we are shown the overlapping sweeps tend to cluster in the genome. Complex patterns of the sweep sharing across populations and continents. Overall, out of the 1,803 intervals shared across populations, 521, which is 29%, were shared across continents, frequently between Europe and South Asia. This is consistent with observed lower genetic differentiation relative to other continent comparisons. An example of a 10 MB window on chromosome 17 with multiple overlaps across many populations. You can see this on the chart to the left-hand side for population codes and also the distributing of sweep interval counts in 10 MB windows across the genome for a single population. This graph shows the enrichment of shared sweeps across the population pairs. For all within continent population pairs, all but 4 out of the 75 Eurasian between continent pairs, the degree of sweep sharing was higher than the background rate, suggesting that the sweep sharing we observed was not driven purely by the haplotype comparisons across closely related populations, which is compatible with more widely shared sweeping haplotypes being broken down by recombination over time. There was a borderline significant correlation between the nucleotide diversity of a sweeping haplotype and the number of populations sharing a sweep. You can see this in this graph because the squares below diagonal representation the null fraction of overlap shared across the population pair. In this slide, you're able to see the signatures of positive selection at the ADH locus on the chromosome 4. Overlapping and shared sweeps enrich the ethanol oxidation pathway. This is a direct effect to how alcohol dehydrogenase oxidizes ethanol to acetaldehyde, a process that is thought to occur primarily in the liver. These data suggest a similar mechanism is at play in individuals of West African ancestry as in East Asians, where the selected allele increases ADH enzyme activity. This results in an adverse physical response to alcohol consumption and the reduced risk for alcohol dependence. So what are the results of all these comparisons? Well, only certain populations have genetic adaptations that make it harder for them to process alcohol, but there's no telling how fast they'll spread to other populations. Analyzing the genomes, the researchers found one of our genetic sites, the alcohol dehydrogenase, which is the ADH gene cluster, showed signs of adaptation. Parts of East Asian ancestry and West African have a genetic variation that makes it uncomfortable to drink too much alcohol. This variation reduces the function of the gene that converts acetaldehyde into the less toxic acetate, thus proving that evolution may make it harder for certain humans of ancestry background to hold their liquor. Thanks for watching. I hope you found this as interesting and informative as I did.